We're privileged today to have Dr. Naomi Bramhall give us a talk from Portland, Oregon. She's going to speak on what animal models and military veterans can tell us about cochlear synaptopathy. She's at the VA and also in the head neck department at Oregon Health Science Center. So Dr. Bramhall, thank you very much for joining us, and please go ahead.
Okay, thank you for having me this morning. So first, just a quick disclaimer, the contents of this presentation do not represent the views of the U.S. Department of Veteran Affairs or the United States government. So prior to 2009, we had a number of assumptions in the field about noise-induced hearing loss. First, that outer hair cells are the most vulnerable part of the auditory system to noise, that if auditory thresholds are not permanently changed, there's no permanent damage to the auditory system, and that hearing conservation programs can detect early signs of noise damage with an annual audiogram. And these assumptions were challenged in 2009 when Sharon Kajawa and Charlie Liberman published this seminal paper describing a mouse model of what later became known as cochlear synaptopathy. So in this study, mice were exposed to an 8 to 16 kilohertz band of noise at 100 dB SPL for 2 hours. And following the noise exposure, they did some physiological measurements in the mice. So they measured distortion product otoacoustic emissions, or DPOAEs, as a way to assess outer hair cell function. So here on the bottom, we have frequency. This shaded region is the noise exposure band that the mice were exposed to. And then on the y-axis, we have threshold shift. So this green 0 line would indicate that no threshold shift had occurred. So if we look at the mice 1 day after the noise exposure shown on this red line, you can see that at and above the noise exposure band, there's a sizable threshold shift, but that over time this recovers. So at 8 weeks, shown in the black, the thresholds have returned to normal. So this is by definition a temporary threshold shift. They also measured auditory brainstem response wave 1 amplitude, the EBR. And this provides an assessment of auditory nerve function. So here we have growth function. So we have stimulus level on the x-axis, wave 1 amplitude on the y-axis. This green line shows data from the control mice, and the red line shows data from mice 1 day after the noise exposure. So you can see that there's a dramatic reduction in wave 1 amplitude. And this does recover somewhat over time, but at 8 weeks, shown here in the black, after that noise exposure, you can see that there's still a sizable reduction in wave 1 amplitude at these higher stimulus levels. So auditory thresholds are normal, but input to the central auditory system is reduced. And this has also been termed hidden hearing loss because it won't necessarily show up on the audiogram. So when they looked at histology in these mice, they saw a loss of the cochlear synapses, which are the synapses between the inner hair cells and the afferent auditory nerve fibers. So here we have cochlear frequency on the x-axis, and basically the number of those cochlear synapses on the y-axis. So the green line shows the data from the control mice. Again, the red line shows mice 1 day after the noise exposure. So you can see in these frequencies above the noise exposure band, there's a large decrease in cochlear synapses, and that doesn't recover over time. So if we look in the black at the mice 8 weeks after the noise exposure, you see the same thing. So noise exposure results in an immediate loss of inner hair cell auditory nerve fiber synapses. And this has been demonstrated in not only mice, but also other rodent species. So they also have shown noise-induced cochlear synaptopathy in rat, gerbil, chinchilla, and guinea pig. And it's also been demonstrated in non-human primates. So this is data from the rhesus macaque monkey. So down here at the bottom, we have cochlear frequency. This gray shaded region shows the noise exposure band. And then on the y-axis is synapse survival expressed as a percentage. So here they're assuming that the control monkeys have 100% synapse survival. So they're plotted up these who have temporary threshold shift after the noise exposure, which are shown in the teal color. you can see that at and above the noise exposure band, they show a significant reduction in synapse survival. And this suggests that noise-induced cochlear synaptopathy occurs in primates, but I want to point out that there's some differences in the noise exposure intensity and duration required to induce the synaptopathy between the mouse and the rhesus monkey. So in the mouse, exposure to 100 dB/SPL band of noise for 2 hours resulted in a temporary threshold shift in synaptic loss. Whereas in the rhesus monkey, to get the same outcome, they had to be exposed to 108 dB/SPL band of noise for 4 hours. So this suggests that primates may be less susceptible to noise-induced synaptopathy than mice, and this is potentially good news for humans. We also know that aging leads to synaptopathy. So these are data from mice across a variety of ages. So here we have lifespan expressed as a percentage on the x-axis and survival expressed as a percentage on the y-axis. And if we look at this line here, which shows survival of those cochlear synapses, you can see that there's a fairly steady loss of cochlear synapses as the mice age. We see something similar if we look at human temporal bones. So here we have age at death on the x-axis and the number of those cochlear synapses between the inner hair cells and the auditory nerve fibers on the y-axis. And you see a very similar decline with age. So this translates to about a 7% decrease in synaptic connections per decade of life. We also know that ABR wave 1 amplitude decreases with age. So these again are data from mice of a variety of ages. So here we have stimulus level on the x-axis, ABR wave 1 amplitude on the y-axis, and the different symbols show mice of various ages. So these are the youngest mice up here at 4 weeks of age, and then the mice get progressively older, and these are the oldest mice at 128 weeks of age. So you can see that the wave 1 amplitude decreases as the mice get older. And we see something similar if we look in humans. So here I've summarized data from 5 different human studies that measured ABR wave 1 amplitude for adults of a variety of different ages. And here we've used regression analysis to statistically adjust these data for sex and average pure tone thresholds. So you see a fairly consistent age effect across the different studies. So let me just give an overview here of what we've talked about so far in terms of cochlear synaptopathy. So cochlear synaptopathy refers to the loss of the synaptic connections between the inner hair cells and the afferent auditory nerve fibers. In animal models, synaptopathy occurs in response to noise exposure, aging, and ototoxic drugs. And while auditory thresholds appear relatively insensitive to cochlear synaptopathy, suprathreshold ABR wave 1 amplitude is sensitive to synaptopathy in animal models. Noise-induced cochlear synaptopathy has been demonstrated in multiple animal models, including non-human primates. However, vulnerability to noise-induced synaptopathy varies between species, and primates appear less vulnerable. And temporal bone and physiology studies indicate that age-related synaptopathy occurs in humans. So this leaves us with a number of unanswered questions about cochlear synaptopathy. Does noise-induced cochlear synaptopathy occur in humans? What are the perceptual consequences of synaptopathy? And can non-invasive physiological measures be used to diagnose synaptopathy? So I'll talk about each of these questions today. We can start with the first question. Does noise-induced cochlear synaptopathy occur in humans? So first of all, there's a number of challenges when it comes to investigating noise-induced synaptopathy in humans. So there's currently no method of confirming synaptopathy in live humans. It isn't ethical to noise expose humans for a research study. Humans are not very reliable at reporting their noise exposure and are exposed to a lot of different types of noise. Unlike mice, humans are not genetically identical, so they may have differences in their susceptibility to noise damage. And physiological measures of synaptopathy, such as ABR wave 1 amplitude, may be impacted by outer hair cell loss, making the results difficult to interpret. So despite these challenges, we can still try and investigate this question. One way that we can investigate it is by looking at human temporal bone data. So this is data from a human temporal bone study. And here they divided their temporal bones into 4 groups based on age at death. They also subdivided these 2 older groups based on noise exposure. So they identified 2 groups shown in the red and the pink of individuals with significant noise exposure. These are the individuals that they identified as having noise exposure. So you can see that they're all male, and they either served in the military or they had a noisy occupation. So if we compare the 2 sort of middle-aged groups, the noise exposed one in the red and the one without the noise exposure in the gray, we have frequency here on the x-axis and survival of those synapses expressed as a percentage on the y-axis. You can see that there's a reduction in synapse survival for that noise exposed group compared to the not noise exposed group. And the pink shading is just meant to demonstrate that reduction. And you can see that that reduction is fairly consistent across frequencies. So there isn't a clear frequency effect of that noise exposure. We can also try and answer that question by using physiological measures in live humans. So this is data from a study that I conducted in a sample of young veterans and non-veterans with normal audiograms, where I measured ABR wave 1 amplitudes. And I subdivided the sample into 4 groups shown down here at the bottom based on their veteran status and their self-reported lifetime noise exposure. So here I've just numbered the groups based on their mean reported noise exposure. So this group, the non-veteran controls, reported the least noise exposure, followed by veterans with low noise, and then non-veterans who had ever used a firearm, and then veterans with high noise reported the most lifetime noise exposure. So these waveforms here are grand average ABR waveforms, and what we're really interested in here is wave 1 of the ABR, and we measured this as the difference between the peak and the following trough. So if I stick these numbers up here, you can see that the group with the least reported noise exposure had the highest wave 1 amplitude, and then wave 1 amplitudes systematically decrease as reported noise exposure increases. But we wanted to confirm that this apparent noise exposure related reduction in ABR wave 1 amplitude was not just due to differences in sex or outer hair cell function between our groups. And so we used a Bayesian regression analysis to statistically adjust for DPOE levels and sex. So the results of that analysis are plotted here. So each of these panels shows a different ABR tone burst frequency that we measured. And then on the y-axis, we have wave 1 amplitude compared to the non-veteran control group. And then the 3 remaining noise exposure groups are shown with the different symbols on the plot. So to interpret this plot, if the symbol is plotted above the 0 line, that indicates that the mean wave 1 amplitude for that group was greater than that of the controls. If the symbol is plotted below the 0 line, that indicates that mean wave 1 amplitude for that group was less than that of the controls. And then this bar shows the 95% Bayesian confidence interval. So if we look at the 2 groups with the greatest self-reported noise exposure, so the non-veteran firearms group in the blue and the veteran high noise group in the black, you can see that across frequency, they show a reduction in mean wave 1 amplitude compared to the controls. So noise exposure related reductions in wave 1 amplitude are consistent with animal models of cochlear synaptopathy. So we don't have to confine ourselves just to looking at ABR wave 1 amplitude. There are other physiological measures that have been shown to be sensitive to synaptopathy in mice. One of those is the envelope falling response, or the EFR. And the EFR is basically a measure of how well the auditory system codes an amplitude modulated stimulus. So this is an example of a sinusoidally amplitude modulated tone. And this is an example of the auditory system's coding of that stimulus. And the better the auditory system does at coding the amplitude modulation, the higher the EFR amplitude or magnitude. So these are data from a mouse study that measured EFR amplitude. So here we have stimulus level on the x-axis, EFR amplitude on the y-axis. And there's two groups of mice. The control mice are shown in the black. And then in the red, we have data from mice with histologically confirmed noise-induced cochlear synaptopathy. So you can see that there's a clear reduction in EFR amplitude for the mice with synaptopathy. So we can measure the EFR in humans as well. The setup is quite similar to the ABR. And so in a second study of young veterans and non-veterans with normal audiograms, we measured EFR magnitude. So here we have 3 different groups, non-veteran controls shown in this light gray color, veterans with medium noise exposure shown in the dark gray, and veterans with high noise exposure shown in the black. And down at the bottom, we have modulation depth. These are shallower amplitude modulations. And then on the other side, deeper modulations. And then we have EFR magnitude on the y-axis. So the thin lines here show data from individual participants, and then the thick lines show the group means. So if we look at the group means, we see a reduction in EFR magnitude for the veteran high noise group compared to the non-veteran control group across the 3 modulation depths. But once again, we wanted to confirm that this is not just due to differences in sex or out of rehearsal function between our groups. And so we used a Bayesian regression analysis to statistically adjust for sex and average DPOE level. So the results of that analysis are shown here, plotted separately for males and females. And we have modulation depth on the x-axis, fitted or modeled mean EFR magnitude on the y-axis. And the symbols here basically show the modeled mean EFR magnitude for each noise exposure population, and the bars show the 90% Bayesian confidence interval. But what we're really interested in is the contrast between these different noise exposure populations, specifically the contrast between the non-veteran controls in the light gray and the veterans with high noise exposure in the black. And fortunately, one of the nice things about a Bayesian analysis is that the output of the analysis is a probability distribution. So we have a posterior probability distribution for the non-veteran controls, and we have a probability distribution for the veteran high noise. And we can contrast these probability distributions. And that's basically what we've done here. So this is a probability distribution that represents the dB difference in population mean EFR magnitude if we were to take the probability distribution for the non-veteran controls and then subtract the probability distribution for the veteran high noise population. So values here that are greater than 0, which is shown with the dark gray, indicate the case where the mean EFR magnitude was higher for the non-veteran controls than the veterans with high noise. And that represents 95% of this distribution. And so we can say that we have 95% certainty that the veteran high noise population has a lower mean EFR magnitude than the non-veteran controls. And we can take this value of 95% and stick it up here on the plot. And we can calculate similar values for the other modulation depths and for the females at the various modulation depths. And we can say that overall across modulation depths and with males and females, we can say with fairly high certainty that the mean EFR magnitude is lower for the veterans with high noise exposure compared to the controls. So noise exposure related reductions in EFR magnitude are also consistent with animal models of cochlear synaptopathy. Now there's a third physiological measure that has been identified as being sensitive to synaptopathy in mice, and that's the middle ear muscle reflex, or the MEMR. And this is also known clinically as the acoustic reflex. And this schematic just shows the contralateral middle ear muscle reflex pathway. So basically sound comes into the ear, goes through the middle ear, cochlea, the auditory nerve to the cochlear nucleus, crosses over to the contralateral superior or olivary complex, activates the facial nerve nucleus, and then that leads to a contraction of the stapedius muscle, resulting in a change in compliance in the eardrum. So you can imagine that if we were to disrupt this connection between the cochlea and the auditory nerve due to cochlear synaptopathy, that would have a negative impact on the reflex. And this is in fact what's observed in a mouse model of synaptopathy. So here we have a elicitor level. This is basically stimulus level on the x-axis. This is a measure of MEMR magnitude here on the y-axis. And then in the black, we have data from the control mice, and in the red, data from mice with histologically confirmed noise-induced cochlear synaptopathy. So you can see that the mice with synaptopathy have a weaker reflex, and the growth of that reflex is shallower. So we can measure the MEMR in humans. In fact, the acoustic reflex is already part of the standard audiological test battery. However, there's some differences in how the acoustic reflex is measured clinically versus how it was measured in the mouse model. So clinically, we use a 226 Hertz probe tone, whereas in the mouse study, they used a wideband probe. And so this figure is just meant to illustrate some differences in the information that you get with these two different types of probes, and how that might impact your conclusions. So these are two plots from two different human subjects where we measured their MEMR using a wideband probe. And because we used a wideband probe, we have information across a variety of different frequencies shown on the x-axis. And on the y-axis, we basically have a measure of the MEMR magnitude. And then the blue shading shows the different elicitor levels that were used. So you can see that as elicitor or stimulus level increases, we get a bigger negative deflection, a bigger reflex. And if we were to compare these two people with this information, it's pretty clear that this person has a bigger reflex and this person has a smaller reflex. But if all we had was the information, if all we had was the information that you would get from using a 226 Hertz probe tone, so if we were using a 226 Hertz probe tone, the only information we would have would be the information that's shown here in these black rectangles. And if that was the only information that we had, we would come to the conclusion that this person had a smaller reflex and that this person had a bigger reflex. So this is just meant to illustrate that the amount of information that you have differs depending on the type of probe that you use, and it may impact your conclusions. So in my second study of young veterans and non-veterans with normal audiograms, we measured the wideband MEMR magnitude. So here we have a elicitor level on the x-axis, MEMR magnitude on the y-axis. And if we look at the group means, we see a reduction for the veteran high noise group compared to the non-veteran controls at the higher stimulus levels. For this analysis, we did not statistically adjust for sex because there's really no evidence in the literature that there are sex differences in terms of MEMR magnitude. And we also didn't adjust for DPOE level because we did a separate analysis that suggested that in individuals with normal audiograms that the DPOE level doesn't impact the MEMR magnitude. But we did do some modeling of the contrasts between groups. And so that modeling is shown here. So here we have a elicitor level on the x-axis, modeled difference in MEMR magnitude on the y-axis, and at the bottom are the different group contrasts. So I'm just going to concentrate on this contrast here shown in the black triangle, which is the contrast between the veterans with high noise exposure and the non-veteran controls. So if we look at that contrast, you'll see that there's very little contrast at the lower elicitor levels, but there's more of a contrast at these higher elicitor levels, such that the veterans with high noise exposure show a reduction in MEMR magnitude compared to the non-veteran controls. So that's shown here, but I'll also point out that the confidence intervals are quite broad and overlap 0. Which suggests that a small increase in MEMR magnitude for the veteran high noise population compared to the non-veteran controls is also possible. So model estimates indicate a reduction in MEMR magnitude for veterans with high noise exposure compared with controls, but the confidence intervals are broad. So I've talked quite a bit about some evidence for reduction of physiological indicators in veterans with high noise exposure. However, there are several human studies of noise-induced synaptopathy that have not found a clear relationship between physiological indicators of synaptopathy, so ABR, EFR, and MEMR, and noise exposure history. And most of these studies consisted of noise exposure groups with a history of recreational noise exposure, including frequent attendance of live music events, attending nightclubs, and playing in bands. So I want to talk about some potential reasons for the differing results across these studies, because I think it's confusing to people why I would see reductions in these physiological indicators for veterans, but then these recreational noise studies would not. So first of all, humans may be less vulnerable to noise-induced synaptopathy than rodents. And so it's possible that common sources of military noise exposure from things such as firearm use and exposure to explosions are just more likely to cause synaptopathy because the intensity level is so high compared to common sources of recreational noise exposure from things like concerts. ABR wave 1 amplitude, EFR, and MEMR may not be as sensitive to human synaptopathy, especially when outer hair cell dysfunction is also present. So it's also possible that both recreational and military noise exposure do lead to synaptopathy, but that military noise exposure just leads to more synaptopathy, and so it's easier to detect. Noise exposure metrics are variable and don't predict susceptibility to noise damage. And control groups may differ across studies. So some control groups may inadvertently include individuals with synaptic or outer hair cell loss, particularly if they've not excluded individuals who've ever used a firearm. And then the clinical MEMR may be less sensitive to synaptopathy than the wideband MEMR. So let me just summarize what I've talked about so far in terms of evidence for noise-induced synaptopathy in veterans. After adjusting for differences in DPOAEs and sex, non-veterans with firearm use and veterans who report high levels of military noise exposure show reduced ABR wave 1 amplitudes and EFR magnitudes compared to non-veteran controls. Model estimates suggest that veterans with high self-reported noise exposure have weaker wideband MEMRs than non-veteran controls with minimal noise exposure, but confidence intervals indicate that stronger MEMRs for veterans with high noise exposure are also possible given the data. Overall, these results are consistent with animal models of synaptopathy and suggest that noise-induced synaptopathy occurs in humans. So that brings us to the second unanswered question: what are the perceptual consequences of synaptopathy or deafferentation? So I've introduced a new term here, deafferentation, which I'll define as the loss of afferent input to the central auditory system through damage to the inner hair cells, cochlear synapses, or the spiral ganglion cells. These three types of damage cannot be differentiated physiologically, and they should have similar functional impacts. So we'll refer to them collectively as deafferentation. Predicted perceptual consequences of deafferentation include tinnitus, problems with speech and noise perception, and hyperacusis. So I'll talk about the first two predicted perceptual consequences today. So we can start with tinnitus. So in my first study of young veterans and non-veterans with normal audiograms, many of the veteran participants reported tinnitus. And so these are results of a Bayesian regression analysis where we evaluated the relationship between wave 1 amplitude and the probability of reporting tinnitus after adjusting for sex and DPOE levels. So the blue dots down here at the bottom show the adjusted wave 1 amplitudes for non-veterans and veterans without tinnitus. And the blue dots at the top show the adjusted wave 1 amplitudes for veterans with tinnitus. And the black line shows the predicted probability of reporting tinnitus for a given wave 1 amplitude. So you'll see that as wave 1 amplitude decreases, the probability of reporting tinnitus increases. In my second study of young veterans and non-veterans with normal audiograms, we measured not only ABR wave 1 amplitude, but also EFR and MEMR magnitude. So these are the ABR data. So we have stimulus level on the x-axis, ABR wave 1 amplitude on the y-axis, and divided the sample into 3 groups: non-veteran controls shown in the blue, veterans without tinnitus shown in the black, and veterans with tinnitus shown in the red. So if we look at the means, the group means, we see a reduction for the veteran tinnitus group compared to the non-veteran controls across all stimulus levels. These are the EFR results. So here we have modulation depth on the x-axis, EFR magnitude on the y-axis. And if we look at the group means, again, we see a reduction for the veteran tinnitus group compared to the non-veteran controls across all modulation depths. And then finally, we have the MEMR results. So here we have elicitor level on the x-axis, MEMR magnitude on the y-axis. And again, we look at the group means for those higher stimulus levels, we see a reduction for the veterans with tinnitus compared to the non-veteran controls. So when looking at the raw data, we see a reduction on all of these measures, suggesting that some forms of tinnitus are associated with deafferentation. However, once again, we want to confirm that this is not just due to differences in sex or auditor self-function between our groups. And so here we used Bayesian regression analyses to statistically adjust for sex and outer hair cell function. And specifically, I'm going to show you the modeled magnitude contrast for the veterans with tinnitus versus the non-veteran controls. So these are the ABR model results. So we have stimulus level on the x-axis. On the y-axis, we have the ABR wave 1 amplitude contrast between veterans with tinnitus and non-veteran controls. So if the symbols are plotted on the 0 line, that basically indicates no contrast, no difference between those two populations. If the symbol is plotted below the 0 line, that indicates a reduction in amplitude for the veterans with tinnitus compared to controls. So here, just at 90 dB, we see a modest reduction in magnitude for the veterans with tinnitus compared to controls, but the confidence interval overlaps to 0. These are the EFR results. So here we see a very clear reduction across all modulation depths for veterans with tinnitus compared to non-veteran controls. And the confidence intervals do not overlap 0. And then these are the MEMR magnitude results. So here we have a elicitor level on the x-axis, MEMR magnitude contrast on the y-axis. You see that there's really no difference between the two noise exposure populations in the for the lower elicitor levels, but as we get to higher elicitor levels, we see a reduction for the veterans with tinnitus compared to controls. So the dots basically show our best estimate of the contrast between those two populations, while the confidence intervals show the plausible values given the data. So you can see that the confidence intervals overlap 0 here. So that indicates that it's possible that there could be a small increase in magnitude for the veterans with tinnitus compared to non-veteran controls given the data. So tinnitus-related reductions in the ABR, EFR, and MEMR measures suggest that some forms of tinnitus are a perceptual consequence of synaptopathy or deafferentation. And I'm going to talk a little bit more about these EFR magnitude results, since these show the clearest reductions for the veteran tinnitus population. So here we see roughly a 2.5 dB reduction in EFR magnitude, but it's not really clear what that means conceptually. And so to place that magnitude reduction into context, we can compare to age-related changes in EFR magnitude. So we know that humans experience age-related synaptopathy, so we would expect age-related changes in EFR magnitude. So these are data from 3 different human studies that measured the EFR magnitude in adults across a variety of ages. And so here we've used regression analyses to adjust these data for sex and average pure tone thresholds. And so if we average the age effect observed in each of these 3 studies, we see that on average, there's about a 1.15 dB reduction in EFR magnitude per decade. And on the previous slide, I showed you the model results that suggested a mean reduction in EFR magnitude for veterans with tinnitus of 2.4 dB compared with the non-veteran controls. And this suggests that the change in the EFR magnitude for veterans with tinnitus is roughly equivalent to 21 years of aging. So these are all very rough estimates, but put this way, it's clear that this reduction in EFR magnitude that we're observing for veterans with tinnitus has a clinically important impact. So I just want to point out that there are also some other studies that have found similar relationships between physiological indicators of deafferentation and tinnitus. So this is a study that also looked at MEMR magnitude here for 2 different groups, a control group shown in the gray and a tinnitus group shown in the red. And you can see they observed a reduction in MEMR strength for the group of tinnitus compared to controls. This is a study that measured EFR magnitude in a control group shown in the black and a tinnitus group shown in the white, and also saw a reduction for that tinnitus group. So I've shown you some evidence that some forms of tinnitus are associated with reductions in physiological measures of deafferentation. But you might be wondering, well, why would tinnitus result, or why would deafferentation result in tinnitus? And in order to understand this, it's important to be familiar with the central gain hypothesis. I'm just going to go over that real briefly. So the central gain hypothesis basically says that if you have decreased peripheral auditory input, this could be due to middle ear issues, outer hair cell loss, inner hair cell loss, synaptic loss, neuronal loss, that this gives rise to hyperactivity in the central auditory system, and that it's this hyperactivity that leads to the perception of tinnitus and or hyperacusis. So the next question then is, well, does deafferentation lead to central gain? And in trying to answer that question, we can first look at a mouse model. So Chambers et al. Treated mice with the neurotoxic drug wabane, resulting in approximately 95% loss of spiral ganglion cells. And the damage increased with time. So there was more damage 30 days after treatment than at 7 days after. So first they looked at auditory nerve function in these mice. So they're measuring ABR wave 1 amplitudes at a variety of different sound levels. In the light gray here, we can see data from the control mice. In the dark gray, we see data from mice 7 days after the drug treatment. You can see ABR wave 1 amplitudes are greatly reduced. And then at 30 days after the drug treatment, shown in the black, you can see that wave 1 amplitudes are even further reduced, reflecting the fact that there's even more spiral ganglion cell damage at that point. They were also able to measure firing in inferior colliculus using multi-unit recordings. So here we have sound level on the x-axis and neuronal firing rate on the y-axis. So this plum color shows the data from the control mice. In the lavender, we have data from mice 7 days after the drug treatment. So you can see that firing is greatly reduced. And then interestingly, at 30 days after the drug treatment, shown in the blue, you can see that firing has increased, even though we know that there's more spiral ganglion cell damage at that point. So this increase as we go from 7 days after the drug treatment to 30 days after the drug treatment is indicative of central gain occurring over time. Things get even more interesting if we look in the auditory cortex. So here we have sound level on the x-axis, neuronal firing rate on the y-axis. The brick red color shows the data from the control mice. In red, we have the data from the mice 7 days after drug treatment. There's very little firing. But then if we look at the peach, this is the data from the mice 30 days after the drug treatment. You can see that firing has increased. In fact, it's increased so much that at these higher stimulus levels, it actually surpasses the firing seen in the controls. So this is basically central gain out of control. So yes, deafferentation can lead to central gain, and this can exceed normal levels of activity. So in humans, unfortunately, we cannot make those same types of multi-unit recordings in the inferior colliculus or the central auditory or the auditory cortex, but we can use electrophysiology to tell us something about central auditory function. So the first part of this waveform should look familiar. This is the auditory brainstem response. And we've already talked about how wave 1 of this response can tell us something about auditory nerve function. If we go further out in latency, we get to the middle latency response, or the MLR. And this is thought to be generated by a combination of the medial geniculate body, the inferior colliculus, and the primary auditory cortex. And if we go further out in latency, we get to the late latency response, or the LLR. And this is thought to be generated by the primary auditory cortex and beyond. So in my second study of young veterans and non-veterans with normal audiograms, we measured the MLR and the LLR simultaneously in response to a click stimulus. And this is a way to get some information about what might be happening in the inferior colliculus and the primary auditory cortex. So these are the MLR results across a variety of stimulus levels. So we have MLR area, which is basically a measure of MLR magnitude on the y-axis. And we've divided the sample into 3 groups: non-veteran controls in the red, veterans without tinnitus in the black, and veterans with tinnitus in the red. So if we look at the group means here, we see a reduction in MLR magnitude for the veterans with tinnitus compared to the non-veteran controls. But remarkably, when we look further on in the auditory system at the LLR magnitude, we don't see a reduction. We actually see an increase in LLR magnitude for veterans with tinnitus compared to non-veteran controls. So this sort of flipping pattern where we go from a reduction earlier on in the auditory system to an increase later on in the auditory system is suggestive of central gain occurring in these veterans with tinnitus. So once again, we wanted to confirm that this pattern that we're seeing is not just due to differences in sex or outer hair cell function between groups. So we used a Bayesian regression analysis to statistically adjust for sex and DPOAE level. So the results of that analysis are shown here, plotted separately for the MLR and the LLR. So after the adjustment, the contrast between groups are more modest, but we still see the same pattern. So we still see a reduction in MLR magnitude for veterans with tinnitus compared to controls, but then an increase for veterans with tinnitus compared to controls for the LLR. The fact that we still see this pattern suggests that deafferentation contributes to the central gain. I just want to point out some similarities between our MLR and LLR results in that deafferentation mouse model that I showed you earlier. So in the mouse model, in the inferior colliculus, at 30 days post-treatment, firing rates were reduced compared to controls. If we look at my MLR data, we see a reduction for veterans with tinnitus compared to non-veteran controls. In the auditory cortex, in the mouse model at 30 days post-treatment, firing rates were similar to controls for lower stimulus levels and surpassed controls at higher stimulus levels, indicating excessive central gain developing over time. And if we look at my LLR results, we see an increase in LLR magnitude for the veterans with tinnitus compared to the controls. So in our measurements that are coming in part from the inferior colliculus, we see a similar reduction to the mouse model in our measurements coming from an area coming from primary auditory cortex. We see a similar increase as what they saw in the mouse model. And so this suggests that our veterans with tinnitus are experiencing central gain in response to deafferentation. So let me summarize what we've talked about in terms of the perceptual consequences of deafferentation as they relate to tinnitus and central gain. Reductions in AVR wave-one amplitude, EFR magnitude, and MEMR strength among individuals with tinnitus suggest that deafferentation is associated with tinnitus. MLR magnitude in response to a click is reduced in veterans with tinnitus, while LLR magnitude is increased even after adjusting for sex and DPOAEs. These results suggest that noise-related deafferentation among individuals with clinically normal hearing impacts the central auditory system, eventually resulting in hyperactivity or central gain at the level of the auditory cortex. And in some people, this central gain is associated with tinnitus. So that brings us to the second predicted perceptual impact of deafferentation, which is stability with speech perception and noise. So let's look first at some animal studies. So obviously we can't measure speech perception and noise in animals, but we can measure tasks that should tap into similar tasks that should be necessary for speech perception and noise. So for example, we can do tests of temporal processing, such as gap detection, or look at tone and noise or signal and noise detection. So this study measured gap detection in chinchillas with approximately 70% drug-induced inner hair cell loss. So here we have stimulus level on the x-axis. On the y-axis, better performance is toward the bottom, poor performance toward the top. And if we look at the chinchillas before and after inner hair cell loss, you can see that following inner hair cell loss, they have poor gap detection, so poor temporal resolution. In this second study, they measured signal and noise detection for rats with presumed noise-induced synaptopathy. So here we have stimulus frequency on the x-axis. On the y-axis, better performance is toward the top, poor performance toward the bottom. And if we look at their performance before and after presumed noise-induced synaptopathy, we see poorer performance following the noise exposure. And this last study measured tone and noise detection for mice with approximately 70% drug-induced spiral ganglion cell loss. So here on the x-axis, better performance is toward the bottom, poor performance toward the top. This is how the mice did for the drug treatment, and this is how they did following drug-induced spiral ganglion cell loss. So following spiral ganglion cell loss, their performance was poor. So taken together, these animal studies suggest that there are some deficits in temporal processing and signal and noise detection following deafferentation that we would expect to negatively impact speech perception and noise in humans. So this is a summary of human studies of deafferentation and speech and noise perception. So each of these studies measured a physiological indicator of deafferentation, but you'll notice that the exact measure that was used varies from study to study. And then these studies compared that physiological measure to some sort of speech perception measure. And you'll see that the speech perception measures vary even more than the physiological measures. So this makes it quite difficult to compare results from one study to another. In this last column, I've indicated the result. So if the study found a relationship between a physiological measure and a speech measure, then that's indicated in green. If no relationship was observed, that's indicated in black. So you'll see that there's a lack of consensus across the studies. But I want to talk a little bit more about this first study. So this is a study that I conducted quite a while ago, before I came to the VA. And in this study, we measured ABR wave-one amplitude and speech perception and noise. And we found, and this was in a sample of adults of a variety of ages with up to a mild hearing loss. And in this study, we found an interaction effect between wave-one amplitude and pure tone thresholds on speech perception and noise. And this figure is just meant to illustrate what that interaction effect means. So here we have wave-one amplitude on the x-axis. On the y-axis is a performance on the speech perception in noise tasks, so better performance toward the bottom, poor performance toward the top. And just for illustrative purposes, we've divided the sample into 6 groups based on their average pure tone thresholds. So here in the blue are the people with the best pure tone thresholds. And then as we get more and more red, pure tone thresholds get poorer. And if we look at the relationship between ABR wave-one amplitude and speech and noise perception, which is indicated by this black line, you can see that in the individuals with really good pure tone thresholds, there's very little relationship. But as the pure tone thresholds get poorer and poorer, that relationship gets stronger and stronger, such that lower wave-one amplitudes are associated with poor performance on the speech perception and noise task. And this suggests that we may be more likely to observe relationships between physiological indicators of deafferentation and speech perception and noise if we look at individuals that have some outer hair cell dysfunction. And this hypothesis is supported by the fact that if I highlight in pink here the studies that included some individuals with outer hair cell dysfunction, either because they included older people or because they included people with some mild hearing loss, that these studies were more likely to show a relationship between physiological measures and the speech measures. So relationships between physiological indicators of deafferentation and speech perception may be more likely when there's some outer hair cell dysfunction. So let me just summarize what we've talked about in terms of deafferentation and speech perception and noise. Animal models of cochlear deafferentation suggest that speech and noise detection may be negatively impacted. Several human studies have suggested a relationship between speech and noise perception and physiological indicators of deafferentation, while others have not. But it would be premature at this point to assume that synaptopathy or deafferentation has no impact on complex speech perception. It's possible that careful test selection is necessary to detect the effects of deafferentation on complex speech perception. And deafferentation may impact complex speech perception more when there's also outer hair cell damage. So that brings us to the final unanswered question: can non-invasive physiological measures be used to diagnose synaptopathy? So observed relationships between physiological measurements and proposed risk factors like noise exposure and perceptual consequences like tinnitus among veterans suggest that these measurements may be useful for diagnosing deafferentation. These measurements cannot distinguish between inner hair cell, cochlear synapse, and spiral ganglion cell loss. However, studies of animals and human temporal bones suggest synapse loss will be the most common type of cochlear deafferentation. In addition, the extent to which these physiological measures are impacted by hair cell damage needs to be explored further. And more than one test measure may be necessary to differentially diagnose deafferentation. And finally, normative values for ABR, EFR, and MEMR measurements will be necessary for diagnosing deafferentation in individual patients. So let me just give you some final conclusions. Physiological measures are consistent with deafferentation among veterans reporting high noise exposure and non-veterans with a history of firearm use, suggesting that noise-induced synaptopathy occurs in humans. Tinnitus is associated with reductions in physiological indicators of deafferentation, and young veterans with tinnitus show electrophysiological evidence of increased central gain in the auditory cortex. suggesting that deafferentation has perceptual consequences. There's some evidence that speech perception noise may be impacted by deafferentation, particularly when outer hair cell function is abnormal, but there's a lack of consensus across studies. And the ABR, EFR, and MEMR show promise as possible future diagnostic indicators of deafferentation, but further work is necessary before they can be used clinically. So I'd just like to acknowledge all the individuals that have contributed to the work that I talked to you about today, and also my funding from the VA RR&D. So thank you for your attention, and I'm happy to take any questions.
Hello, my name is Dr. Kevin Peng, neurotologist here at the House Institute. Thank you for watching this video. The House Institute provides free educational videos for hearing health professionals worldwide. To help support videos like these and other educational efforts, please consider donating by clicking the link in the description box below. Your generous support allows us to keep videos like these at no cost for you and others. Thank you.